The title of the article that we use is Metagenomic Diagnosis of Bacterial Infections, Emerging Infectious Diseases. Here, we stated the authors and the date published of this article. Metagenomics is a rapidly advancing field that enables the study of genetic material recovered directly from the environmental samples, bypassing the need for isolation and cultivation of individual microbial species. This powerful approach has revolutionized our understanding of microbial diversity and function in complex ecosystems. There are two types of sequencing technique, which are high-throughput DNA sequencing and shotgun sequencing. High-throughput DNA sequencing is a technique that rapidly sequences millions of DNA fragments in parallel, allowing for comprehensive analysis of genetic material. In shotgun sequencing of metagenomic samples, DNA from diverse microbial communities is broken into small fragments, sequenced, and then reconstructed computationally. This approach enables the study of microbial diversity and function within complex ecosystems, such as soil or the human gut, without the need for culturing individual species. By identifying genes and species present in the sample, researchers gain insight into community structure, functional potential, and ecological dynamics, advancing our understanding of microbial roles in various environments. In this research, we'll be using the shotgun sequencing. Shotgun sequencing creates a genomic library by ligating random genomic DNA fragments into a vector and then randomly sequencing the clones. Every fragment in the library is partially sequenced. The goal is that there will be some overlap between the parts. The sequencing data is subsequently assembled into cotics by computer, which identify regions of overlap. Closing the gap between cotics can be accomplished by looking for library clones that hybridize two probes from previously identified cotic ends, or by amplifying using primers that anneal to the ends of the two known cotics.
Application for shotgun sequencing in metagenomic. Shotgun sequencing method can be applied in the fields of infectious disease and diagnosis surveillance, microbiome research, antibiotic resistance surveillance, environmental monitoring and bioremediation. Lastly is food safety and quality control. Step 1. Sample preparation. In order to evaluate the effectiveness of high-throughput DNA sequencing in detecting bacterial pathogens, genomic analysis was conducted on fecal DNA obtained from a patient during and following an episode of diarrheal illness. Sequences showing best match for C. jejuni were detected only in the illness sample. This metagenomic approach exhibits potential for the detection of various bacterial species. From the article, the sample has obtained from a 34-year-old man fell ill after eating dinner out with his family. 3 days later, he, along with 2 family members who also eat undercooked chicken that night, experienced severe diarrhea, stomachache, and shivering. The patient's fecal sample was collected 4 days after symptoms appeared and stored at negative 80 degrees Celsius. Conventional culture methods and specific PCR tests for norovirus conducted at a clinical laboratory in Osaka, Japan did not detect any pathogens. High-throughput DNA sequencing was performed on the fecal sample using a GS20 sequencer from 454 Life Science. DNA was extracted from the illness sample using a QIAM DNA stool mini kit. Another fecal sample was collected from the patient 3 months later when he had fully recovered and stored at negative 80 degrees Celsius for DNA extraction and sequencing.
The sequencing used for this experiment is high-throughput DNA sequencing, or specifically next generation sequencing (NGS) was used. NGS eliminates the bacterial cloning step required in traditional Sanger sequencing. Single isolated DNA molecules were amplified and analyzed with massively parallel processing. During analyzing, the BLASTEM program was used to match the obtained sequences with organisms in the database. A classification system was employed to assign the sequences to specific organisms. The analysis enabled the detection and identification of various bacterial pathogens in clinical samples. The advantages of this study is that it highlights the importance of metagenomic studies in identifying unknown virus pathogens and represents a significant advancement in clinical examination for infectious diseases.
The table shows the frequency distributions of the species from which the sequences have close matches. For the recovery sample, sequencing yields 106,327 effective sequences, and for the disease DNA sample, 96,941. The majority of the bacterial species found in both samples belong to the phylum Bacteroidetes, which is the typical gut flora of humans. The frequency of the species in the DNA samples from the sick and recovered subjects did not significantly differ from one another. One notable distinction between the two samples was that whilst no sequences from the recovery DNA sample exhibited any meaningful matches, 156 sequences from the illness DNA sample show past matches for sequences originating from Campylobacter jejuni. Numerous ribosomal RNA and DNA polymerase genes, among other housekeeping genes, were found in the C. jejuni sequences from the deceased DNA sample. As a result, they strongly implied that C. jejuni was present in the disease-related fecal sample, due to the fact that C. jejuni is a bacterium that often is not found in the intestines of healthy people and causes acute gastroenteritis.
Hi, my name is Siti Noshazrina. Next, we go to the part 6, which is validation. Clinically, Campylobacteria are the thermophilic groups such as Campylobacter jejuni, Campylobacter coli, Campylobacter abscelensis, and many more. In this case, the validation to detect the Campylobacter jejuni is by using the real-time polymerase chain reaction, which we can call in short PCR. PCR assay indicates its ability to amplify appropriate target strains or species. A sensitive PCR assay can detect the thermophilic Campylobacteria like Campylobacter jejuni directly in the diarrhea stool. Lastly, we go to the conclusion. In conclusion, metagenomic is the study of the structure and function of entire nucleotide sequence isolated and analyzed from all organisms, which is typically microbes in a bulk sample. The advantage of metagenomics is provide more comprehensive identification of the microorganism present in a sample than other DNA-based techniques. Metagenomic is important as its ability to uncover new and previously unknown microbial species. By using metagenomics, researchers can analyze microbial diversity and identify new protein, enzyme, and biochemical pathways until in the future. That's all from us. Thank you.